I mean, rats have to eat. That's how they do it.
That's not until they're naked.
So here's a real shocker. Most mammals are born without hair, right? I don't know how that's helpful to you, but there it is. So we're going to talk about catecholamines today. That is sort of a large group of neurotransmitters, right? So we've talked a little bit in the past about basics of psychopharmacology. We talked about methods. Starting today, over the next couple weeks, we're going to hit some of the major neurotransmitter groups, right? Again, I don't expect you to know a whole lot of what's going on, but you need to know some basics, right? You need to understand just a little bit of the synthesis pathways, how we can block reuptake, things like that. Where are the medications going to create sort of interactions in this process, right? All right, so there are sort of two large groups of catecholamines. One of those is going to be dopamine. The other is what we call the noradrenergic system. That includes epinephrine and norepinephrine, right? Those guys are in there. All three of these are really related to each other structurally. That's why we put them all in the same group. And so they're going to have some similar action and pathways. All right, there you go. They're your three main guys, right? Sometimes we'll call these guys monoamines. Monoamines are kind of a larger group above the catecholamines that also include some other things, right? For those of you interested, this is the catechol nucleus. All your catecholamines are going to have this fun sort of structure, right? And then, I mean, for you, anybody take biochemistry?
I had organic chemistry.
Wow, that was something.
Yeah.
And you didn't continue on in that field? I'm a little surprised.
No, I didn't.
I mean, once you make it past organic too, it's sort of downhill from there. That's the hard part.
Yeah.
But you bailed out on it anyway, that's fine.
Yeah. That's the part.
Gave up. So this is your amine group, right, NH2. Room full of quitters. What do you guys do? Epinephrine and norepinephrine, just to give you some idea, they're also known as adrenaline and noradrenaline. Why do we call it adrenaline? If you sort of take off the AD and the INE, you're left with renal, right? So all of this stuff is made in your kidneys. You guys know the kidneys, right? Those are those kidney bean-shaped organs. It's a good definition. Jessica, I seriously had this anatomy book that said that the kidneys were the kidney bean-shaped paired structures. And I thought, it was the kidneys first, not the kidney beans, right? We call them kidney beans because they're shaped like kidneys. And then I thought, kidney beans, you would define that as like the kidney-shaped bean, right? I mean, that's the kidney bean. That doesn't make sense. It wasn't a great one. But it's called adrenaline, or noradrenaline. They're made in the adrenal glands, which are kind of a little elf hat that sits on top of the kidney, right?
How are they similar to like endorphins? I had a 201 student ask me that, so that's why I'm asking you.
Really?
Yeah.
So you want me to tell you what the answer I would give them?
Yeah. Because I couldn't give them an answer, but I was like, I'll look into it.
Yeah,
So this
I
is me
mean,
looking into it.
just tell them they're different molecules, they do different things, and that adrenaline and noradrenaline are really associated with sort of increasing the activity in your sympathetic nervous system.
Okay.
I think that would be a good answer to an odd question.
Yeah. Well, because we were talking about endorphins when you're exercising and stuff, and
Yeah.
so they put adrenaline with that.
Yeah, I mean they're going to again be released at similar times, or they're going to do sort of different things. Not a big deal. All right. So adrenaline, noradrenaline, we've got it. You can get hormones out of your adrenal glands. That's fun. Don't worry about this. I mean, it's just telling you like you make drugs, right, in your body. Basically, we want to think about tyrosine hydroxylase. This is really an important enzyme in the synthesis pathway, right, that we're going to talk a little bit about today. All of these guys start as the amino acid tyrosine. Okay, that's one of your 26 amino acids. yeah, that you have, right? So you have a group of amino acids. You guys have heard of these things, right? So we're going to take tyrosine, we're going to make dopamine and so forth. So tyrosine hydroxylase and this guy called AADC, don't worry so much about that. These are going to be found in neurons that make dopamine. Okay, so this is a good marker. You want to know, is that a dopaminergic cell? We're not really going to see if it has dopamine in it, right? And I know that's kind of weird, right? You think, well, we'll just see if there's dopamine. That's tricky, sort of experimentally. But we can definitely tell if it has tyrosine hydroxylase. And we know it has to have tyrosine hydroxylase to make dopamine, so we can fairly safely assume that if it has tyrosine hydroxylase, then it's going to make dopamine. Here's another enzyme, dopamine beta hydroxylase, that makes norepinephrine. So that's how you can kind of tell the difference, right? So if it has one or the other, you'll know which way it goes. Anything that ends in ASE, right? You know that's an enzyme, and that's an enzyme that's going to, like, you know, make changes, right, to the structure. So if you're looking at this sort of the synthesis pathway here for dopamine and norepinephrine, you start with that guy tyrosine. You're going to add you know, your hydroxyl group. You're going to do a few other things to make dopamine, and then you're going to end up with norepinephrine. This guy is DOPA. A lot of you have heard of L-DOPA, right? And we talked about that some. This is that precursor that you would take if you had Parkinson's disease, for example, and you need to increase your amount of dopamine, right? Now, Destiny, this would help you if you didn't also have a problem with your tyrosine hydroxylase, right? So, well, I guess you're bypassing tyrosine hydroxylase, but AADC, right? So if you've got enough AADC to handle this reaction, that's fine. If you don't, jacking up the DOPA levels is not necessarily going to help, right? So you have to make sure that the issue is not enzymatic. Tyrosine hydroxylase is what we call a rate-limiting enzyme. So if we can increase or decrease the activity of tyrosine hydroxylase, then in the normal system, we're going to increase or decrease the amount of dopamine that's available, right? That's that point where you really want to make the change. Okay.
So does L-DOPA contain that? I mean, does it have
No, L-DOPA is basically just providing you with this. So you're bypassing that tyrosine hydroxylase step altogether, and provided you have your AADC enzyme, you'll just keep making dopamine. Folks who have Parkinson's disease, it's not that they're missing out, you know, unnecessarily on any of the enzymes. They're just losing cells completely that typically make dopamine. So if you can sort of bypass, provide that precursor, it still helps. Also, this is something important to keep in mind. I like to tell people, whatever is going to happen in your body at any given time is happening right now. It just might not be happening at a rate high enough to make a difference, right? So you can change the rate of that reaction by providing precursors, right? So you can provide, you know, more on one side of the reaction than the other, and it'll drive the reaction to the other way. Does that make sense? Because it's all about these like random interactions. If you think of, it's really quite amazing that it's just some random interaction with like, you know, tyrosine floating around, and if it's going to like accidentally bump into tyrosine hydroxylase, right? And when they bump into each other, then you're going to turn that into DOPA and go forward, right? So whether or not these actually accidentally interact, that's a, you know, how do you affect that accidental interaction, right? And so in this case, you know, if you want to make dopamine, how do you affect that sort of accidental interaction with the AADC? You can do that by increasing the amount of DOPA available, right? So whatever AADC you have, you're going to be more likely to accidentally bump into that because you're going to increase the amount of DOPA out there. Does that work? How many of you have ever lost a dog? That's a sad story.
I did not see that coming.
I know, right? And it's just a real surprise for you. So let's say you've lost a dog. Okay. So here's your dog. And you want to return that dog home, right? So how might you do that? You might get more of your friends out looking for your dog, right? And so the more of your friends you have, the more likely they are to accidentally run into your dog, and then you're happy. Does that work for you guys?
So why does L-DOPA only work for a limited period of time?
Yeah. There's some real problems with L-DOPA. One is your brain cells are still dying. So you have to like continuously jack up the amount of L-DOPA you're taking, which is fine. Keep doing that until you start hearing voices. Because schizophrenia, and we'll talk about that much later, that's an overabundance of dopamine, right? And so then if you're on L-DOPA long enough and you continue to increase the dose, you will develop schizophrenic-like symptoms. So that's a problem. And you're not going to be able to, at a certain point, once you lose enough cells, it doesn't matter how much of the precursor you provide, right? You're not going to be going forward. And we don't just want to give people squirts of dopamine. We don't want to do that. Because dopamine is it's a powerful, heavy hitter. So you really want to have some bit of control on that process. Kristen?
How do the antipsychotics work in this equation?
Yeah, so we'll talk about that.
Okay.
And they can work in a variety of ways. Typically, a lot of them will not necessarily work in the synthesis pathway. A lot of the medications that you're going to run across are going to work in either blocking reuptake, right? So they won't necessarily because again, you don't just want to give someone, more, but the antipsychotics will be agonists on the dopamine, right? So they'll reduce dopamine. You want to be careful how you do this, right? So you don't want to knock down too much dopamine too fast. You don't want to just completely destroy it all, because then you've developed a movement disorder called tardive dyskinesia, which is essentially drug-induced Parkinson's disease. The problem with tardive dyskinesia, as opposed to most other sort of side effects, like how many of you have ever taken a drug that causes a side effect? What do? You just stop taking the drug, right? It goes away, that's fine. Tardive dyskinesia, unfortunately, does not disappear. Once you've developed it, you've got it. And so there's no way to reverse that, once you've taken too many antipsychotics or for too long. And so you're constantly playing this game, right? Like with dopamine. Like what's the fine line between Parkinson's and schizophrenia? And that's where you want to be. I'm assuming most of you are there naturally. Most people are there naturally, right? Because it's a small percentage of people who have Parkinson's and a small percentage of people who have schizophrenia, right? And so most of the people are in the middle. So you do a pretty good job on that. Now that you're putting the effort into it, it just happens. Does that make sense? But we'll talk about where antipsychotics can have effects. We'll talk about where, like antidepressants or something can have effects as well, which will sort of do the opposite. We should briefly talk about negative feedback, because you're going to get a lot of that in this class. That was just right there for me. I mean, it's low-hanging fruit. Negative feedback in this case, what we mean is, how do we control this process, right? So it doesn't run out of control. And we're going to do that through negative feedback. So if we really have high catecholamine levels, this could be dopamine, norepinephrine, or epinephrine, that's going to inhibit that tyrosine hydroxylase enzyme, right? So it's going to inhibit its activity, so we stop making, in this case, so much of the DOPA, which is a precursor for all of those neurotransmitters. So we'll inhibit that activity, slow the process down until catecholamine levels in general drop down, and then that tyrosine hydroxylase will go back up. It's all about homeostasis and negative feedback. Positive feedback does not work. That's why we never say good things about your work, Jason. In a biological system, positive feedback is bad. Typically, there's only like one time positive feedback works, and that's if you're trying to expel something from your uterus. True story. The only time positive feedback loops work is during birth, right? And it works through oxytocin. So if your uterus gets stretched, it's going to like scream up to your brain like, hey, I need some help. And your brain casts it just in its brilliance. It says, here's some oxytocin. Now, oxytocin is actually involved in smooth muscle contraction. So it will cause your uterus to really squeeze down even tighter. And if you've got something in there like 10 or 15 pounds, right, like a baby, which is how big they are, that's how big they are, I promise you. You've got like a 15-pound baby in there, that's going to stretch your uterus even more, right? And so then your uterus is going to go, hey, brain, that didn't really help. It's worse. I need some help. Do something. And then the brain's like, hey, what about that oxytocin stuff I've got? That's going to be great. And so it'll release more oxytocin and your uterus will continue to like contract. Now the great thing is like typically the baby comes out of the uterus at some point, right? Like 20 to 30 hours later, once this process has started, the uterus or the baby exits the uterus, and then you've got Charles over there instead of uterus or fetus, you know, so you've got a new name. And at that point you're fine because that positive feedback loop ends with expulsion of your offspring, right? It does not work in any other sort of system. If your blood pressure gets too high, and so you decide, you know, it would be great if I just increase my blood pressure a little bit more, and then you go like, wow, man, my blood pressure is even higher, so what should I do? I should make my blood pressure even higher, and then you start like blowing out blood vessels, right? Just positive feedback loops only work if there's a clear end to the process, and there's a clear end to shoving something out of your uterus, right? Kristen's writing that down furiously. So, negative feedback loops, that's what we're going to be dealing with here. That's how we're going to self-regulate. There are a lot of components to negative feedback loops. We'll talk about autoreceptors here in a bit, right? Autoreceptors are going to be part of this process as well. Not a big deal. If the neurons start firing a lot, then tyrosine hydroxylase is going to be stimulated. We're going to accelerate that catecholamine synthesis. That's going to be great. There's this whole thing about phosphorylation. I'm not going to expect you to know that. If you're interested in it, Kyle, it's there for you, okay? But that's probably not going to be something that appears on the test. Here's L-DOPA. We already talked about that, right? That's that precursor to Parkinson's disease. Here's a drug, AMPT, that's actually going to block tyrosine hydroxylase, right? This is something you could administer, which would completely stop the synthesis of any of your catecholamines. Again, that's dopamine, norepinephrine, or epinephrine.
Why would you want to do that?
Well, just to figure out how it works. That's one reason. You wouldn't necessarily do that to a human. There are other reasons. Kristen, why might you want to stop some dopamine synthesis? We just talked about this.
So you don't have schizophrenia?
Yeah. So there's that. You really have to put your catecholamines into vesicles. If you don't put them into vesicles, they'll get destroyed, right? So if they're just out floating around, you know, just for fun, those enzymes are going to come along and just, you know, tear them to pieces. We'll talk about those enzymes in a minute. I haven't really introduced you to those. But typically, we have these guys stored in vesicles, and there's a transporter that does that, vesicular monoamine transporter, right? That's kind of interesting. And it's going to pick up your monoamines. Remember, you know, we were talking about monoamines is kind of a bigger group, but includes these guys, and it's just going to put them into vesicles. Okay, so that's going to be cool. We put them in the vesicles, store them for whenever we need them. You can actually block that storage. There are drugs that do that, right? Which would also prevent the release of dopamine, because we're not going to have it in these nice prepackaged vesicles for you. Here's that transporter just sucking up dopamine into the vesicle. We can release that vesicle when we're instructed to do so, unless we block that process. Not a problem. Hey, this is something. You might also, you know, want to block dopamine or norepinephrine production or storage if you want to cause depression or put someone to sleep. Everybody's interested in that. It's a great way to stay in business. Nobody thinks about that, right? Like if you were a dentist, like give away little Snickers bars.
I think we're quite all right.
You sure?
Thanks.
You guys don't need additional clientele? Okay.
There's a lot of mental health concerns.
Got it.
We're good.
Got it. Hey, who loves rabbits? Yeah, these rabbits are fine. These guys have had their catecholamine production reduced, right? And these guys were given DOPA, or L-DOPA, and they recovered. How about that? What if I tell you these are the same rabbits? Will that make you feel better? So, okay. Let's imagine they are.
Yeah.
You're welcome. So I don't know if they're the same rabbits. They very well may be, but I'm not convinced that they necessarily are. But anyway, that's how they figured out L-DOPA can help you if you have a dopamine, a reduction in your dopamine level. So you can think about these guys as being rabbits with Parkinson's, right? Because they're not necessarily developing or creating enough dopamine, right? And then here we've treated them, right? The nice thing is they'll be fine once we stop, like artificially stopping their catecholamine production.
So they're not dead.
They are not dead.
Okay.
They're just sedated.
They just look very dead.
Well, they're depressed and
Throw me
sedated.
off.
If you're depressed and sedated, you might look dead, right? I mean,
Makes sense.
that's possible, right? So there you go. That's fun. What else do we want to know? Hey, who's heard of amphetamine or methamphetamine? Yeah, that's something, right? When somebody's being arrested because they've got some methamphetamine, when there's like a van that it's got something on the side that doesn't really match the people who are waiting on out on the sidewalks, sitting there while there are like 5 police cars, parked around it. I see this occasionally when I'm driving down US 60. They have people like pulled over at the Boost mobile store. Have you ever seen that? It seems to happen right there. Yeah. So there's this van, and you think, oh, that's a guy who installs windows, but like the 3 people they have out on the like on the side of the road there, they definitely don't install windows, and there are all kinds of police going through that. What's interesting about this is, so when you release catecholamines, it actually leads to behavioral activation, right? So that's what happens. So when you release these catecholamines, it's going to cause some behavioral activity, right? You're going to activate that. So it makes sense, right? So that's cool. There are some drugs like amphetamine or methamphetamine that actually don't require the cell to fire for you to release your catecholamines. So if you think about someone who's on methamphetamine or amphetamine, what's like one of the number one things you're going to look for? Think about that behavioral activation of those individuals, right? These rabbits are not on methamphetamine, right? You can tell because the image is in focus, right? If these rabbits were on methamphetamine, it would just be white blurs, right? That's how you know. I just don't think about these things, do you? No, why not, Kyle? Got other things on your mind. Should have this stuff. So it's going to increase locomotor activity. As you get higher doses, you start doing what are called stereotyped behaviors, right? So with rats, we're thinking about like sniffing, head and limb movements, licking, biting. But you see these in people as well, right? If you think about individuals who are, on high doses of methamphetamine, they'll have stereotyped behaviors. Not a big deal. This behavioral activation really comes because we're increasing dopamine, we're increasing that stimulation of those in the nucleus accumbens and the striatum, right? So the main sort of pathway for drug addiction and behaviors, right, is the ventral tegmental area onto the nucleus accumbens. Like that circuit, that dopaminergic circuit gets keyed up every single time you do something, Olivia, that's rewarding, right? Every time you do something rewarding, that gets activated, okay? And when you do something that's really rewarding, like methamphetamine, it gets really, right? Because we're getting that massive spike in dopamine levels, right?
Is that related to obsessive compulsive disorder at all? Like
Yeah,
dopamine?
that's actually a great question, and I don't necessarily know the relationship with dopamine levels, but obsessive compulsive disorder does involve, and this mentions the striatum, which is part of the basal ganglia, right? There are some folks who are now and have been for a few years looking at the implications of OCD, Tourette's, autism spectrum disorder, and that basal ganglia. We talked a little bit about that, right? And if there's dopamine released in that basal ganglia, which is also where your Parkinson's right? That's the nigrostriatal pathway, so that's coming to the striatum. Those cells dying. You're definitely going to get that stereotyped behavior, right? So when you get activation in there. Hey, so that's a good one, right? Like, how do you know if someone is OCD or if they're on methamphetamine? They might still be, continuously washing their hands. I don't see a lot of people on methamphetamine just like washing their hands, right? They have other stereotyped behaviors, okay? But still, it's the same kind of thing. Right? It's the same source. So that's exciting, right? Making connections here for you. Hey, remember that time, Cassie, I said we'll talk about autoreceptors? You seem really excited about that. So what an autoreceptor is, it's this guy who is going to monitor the release of your neurotransmitters, right? And when it's activated, it's going to go, man, you must have dumped out a bunch of catecholamines. Time to shut down, right? So shut down the release. So they will actually start to inhibit the release of those neurotransmitters. They actually start opening potassium channels. That's probably going to be more than you want to know, but the reason it opens potassium channels is so it can leak out more potassium inside of the cell. Is going to become more negative, and it's actually going to stop firing. So that's kind of fun. It also screws around with calcium influx. Calcium influx is necessary to release vesicles, right? And if you stop the influx of calcium, then you're not going to be releasing your vesicles as much. So that's exciting. Still more autoreceptor business. This is kind of interesting. Cells actually have neurons have two sort of firing modes typically. They have what we call like single burst mode or tonic firing. This is kind of interesting. It's just kind of every now and then you're getting a spike, right? You're getting an action potential. We talked about that a little bit. That'll actually get you what we call tonic release of dopamine. If you have burst mode, right? So this is when you're going to get a bunch of spikes, a bunch of action potentials just jammed together, right? And there's going to really fast, 10 or 20 of these guys, and a little break in 10 or 20. That has what we call phasic release of dopamine. So you get this like massive wave of dopamine release, and then nothing, and then a massive wave again, and then nothing, right? Instead of this sort of like steady sort of release of dopamine with the single spike mode. What's interesting about that is when you do the burst mode, you can actually sort of flood the synapse with dopamine, right? Because those reuptake devices, those enzymes, that process gets overwhelmed, right? That takes too much time. So let's think about like this class. What if I give you one assignment per week? You can manage that, right? But what if I gave you week 1, 15 assignments? You're going to get overwhelmed, right? Because you're getting all of that at once before you have time to take care of it. And that's what happens here with that spike firing. All that dopamine gets dumped out, and your enzymes and your reuptake transporters don't have time to pull all that back. That's exciting. Don't worry too much about this. This is just like dopamine firing in rats in that ventral tegmental area, which is kind of cool. Kind of see after feeding, you get these extra bursts. Hey, the VTA in rats also gets excited when they take care of their kids, like their rat pups. That's interesting, right? I always tell people, like, your mothers don't love you, they're just addicted to taking care of you. Tell your clients that. That's probably not helpful, right? Somebody comes in and it's like your mother never loved you anyway. She was just addicted to taking care of you. We probably don't have to worry about in passant synapses. This is a beautiful diagram. These little kind of bumps, those are all little synapses out coming off of these processes. I don't want to spend too much time talking about receptor subtypes because I don't expect you to remember all of the receptor subtypes. And for another reason, Jessica, probably in 2 weeks there'll be a new receptor subtype, right? They're constantly finding new receptor subtypes, so don't worry about it, right? But we do need to understand what are subtypes, why are there receptor subtypes, and that can actually have sort of important things going on. Sometimes you will hear folks say, well, this is a D2 activator or a D1 activator, or this is a, gap, right? And so you'll need to understand that just because it works on the dopamine system, what receptor subtype it is really important. This becomes very important when we start talking about opioids, because they're like mu and kappa, they do completely different things, right? And so that receptor subtype discussion there is going to be very important, but it's fairly important with dopamine as well. So like your autoreceptor is like a D2, right? So if I said this was a D2 agonist, it was a D2 blocker, for example, and we're going to block those autoreceptors, then that's going to increase the release of dopamine, right? Because the autoreceptor is never going to become activated. If I'm activating the D2 subreceptor, subtype, then we're going to reduce the release of dopamine, right? Because that autoreceptor is going to go, man, you're dumping out a bunch of dopamine, I've got to stop. There's too much coming out. They make mutant mice. They make them by exposing them to radiation. No, that's not how they do this. This is through a series of breeding and genetic things that I don't necessarily expect you to get a handle on. But they will make knockout or knock-in mice, right? Is kind of what we call those. Some of those have different types of receptor subtypes, different profiles, and that can actually cause different, they can be maybe more or less sensitive, in this case to cocaine. It's just the example we give you, right? There are people who have, right, different sort of receptor profiles, right? We've talked about that some. So that's something to keep in mind, right? When you're not pumping these people full of drugs and then they don't get any better. Could be a variety of reasons why. I know that really helped you out, right, Jason? I was like, you thought I was going to give you this major insight, right? Like, wow, I've got a client who's just taking a bunch of medications and they're not getting better. He's going to tell me what's wrong. And I wasn't going to.
Actually, I was thinking one of the articles were about mutant mice this week, but then I was wondering if I did the wrong chapter.
I read that too.
Yeah, okay.
It was about mice?
It was about mutant
Knockout mice.
Good.
Knockout mice.
Yeah.
Yeah, it was
Acetylcholine.
about the cocaine, yeah.
Yeah, cocaine, right?
Yeah,
Cocaine in
that's
mice.
But they mentioned cocaine in it too.
And
Mhm.
acting.
When I read those.
Well, I'm going to make a note of that.
So they're bred that way?
Yeah, intentionally.
It
Forced.
didn't say whether they did that to each individual mouse or if it was bred to be that way.
Yeah, it didn't explain how they were
How they
knockout
became
mice.
knockout mice.
Oh,
It's
so I
just
can
a
tell you how
bizarre.
I can tell you how they become knockout mice pretty easily, right? And you can make knockout humans if you wanted. You can do this, right? Which you should consider. Have we talked about genetic counseling in this class? Like if you wanted to have kids?
Yes.
Yes, that's exactly how you do this. And you just do it with mice, right? Now it might be a little different with mice because you might have to take that first mouse and screw around with its DNA a little bit, right? You might have to inject something or delete something from its DNA. But let's say you have a mouse who has doesn't have your D2 autoreceptor. That could just happen naturally. Let's say it probably didn't. But you're going to have to remove that, from their DNA, and you just breed them, and you just continue to breed these mice, and then you get like a whole litter of them, right? That's all.
Can you do that? Because like my assumption when I was reading it, the implication would be, hey, if you could knock that out, then you could kind of interrupt that for the sake of addiction.
Yeah.
Can you do that like after humans are already like living and stuff? Like I assume that's how they did it. I guess I don't know.
Yeah, I mean you could. I mean the problem is there are a lot of problems there.
Like why are they? What is their hope when they're doing those studies? I guess.
Yeah, I think they're really just looking at mechanism. I mean the problem is if we don't understand the mechanism, then any treatments that might come out, you're not gonna, you're not gonna understand what kind of treatments, like where to target this, right? Or if like, oh, okay, so it is the D2 autoreceptor that's involved. I think a better approach with humans would be to give them an agonist or an antagonist,
Right, yeah.
right? That could alter D2 activity to get whichever way you want it to go on that sensitivity. Making mutant humans is...
Okay, I'm not saying actually making.
It's
my
probably a
thing
little more
is like, can you
complicated.
target that, like you talked
Absolutely,
about?
absolutely, and that's what they're trying to prove here. They're just saying, well, if they don't have these autoreceptors, then here's the result that we've got, and then that's how it's playing a role. Did that make sense? So again, if you stimulate autoreceptors, you inhibit release, not a big deal. So again, withdrawal from opioids, that will actually activate your noradrenergic system, right? And so there are drugs that we will give to folks that are agonists for those autoreceptors, right? So they kind of help that process along. For those of you that have dealt with individuals who are trying to deal with substance use disorder, is that the right term? Again, we've talked about how this can be fatal, right? Depending on what set of symptoms and what drug. And so understanding how to mediate those symptoms, one, will improve sort of the likelihood that you're going to stick with it and not relapse, right? At least in that immediate time frame while you're going through it, because you know, a lot of folks will be like, well, I feel horrible, I've not had any alcohol for 2 days, and maybe to go back to that, or opioids, right? I mean, I've not had any heroin in a while, so I'm going to try that again, because I feel bad, I'm trying to quit. And if you can mediate that effect and ease people down, it's better. Other drugs, if you antagonize, you can block autoreceptors, that'll increase, not a big deal. Blocking those autoreceptors and increasing that norepinephrine release actually sort of provokes withdrawal symptoms and drug craving in folks who are opioid dependent, right? So we're, again, teasing out that sort of mechanism there and the role those play. Here's basically just a graph showing you what I just told you. How are we going to inactivate catecholamines? Again, reuptake. This is one of the primary ways we'll do this. There are reuptake transporters, dopamine transporters. They're just going to reach out and basically pull that dopamine back in to wherever you've released that, right? And you can recycle that dopamine or norepinephrine and use it again. So we'll repackage those, drop them out. This is normal activity. Again, if you have those mice that don't have dopamine transporters, they actually don't respond to psychostimulants like cocaine or amphetamine. That's kind of interesting, right? If you think about it, they're not going to be pulling that dopamine back in. So I'm not going to know if they're on cocaine. Hey, who loves TCAs? No, they don't really prescribe TCAs much anymore, right? Tricyclic antidepressants. You guys don't know, do you? Mm-hmm. The side effect profile for TCAs is pretty nasty, right? And so you're not really going to be seeing folks that are on. Those are kind of the first generation of antidepressants, or tricyclic antidepressants. It's called tricyclic. The structure of it has sort of three.
So, amitriptyline, is that one of those?
I'm not sure if amitriptyline's tricyclic or not. And I definitely can't spell it. Let's see what we've got. Yep, it is a tricyclic. Hopefully we got a picture. See how it's 3 circles? That's how you know. Yeah, and these definitely have, I mean, here we're already talking about side effects: dry mouth, trouble seeing, low blood pressure when you stand up, sleepiness, constipation. That's enough right there. But they can cause seizures, increased risk of suicide, urinary retention, glaucoma. There are heart issues. All kinds of problems.
I was put on that when I was younger for chronic headaches, and I quit taking it because of the side effects.
Yeah, yeah. There are other uses for it besides an antidepressant, but that's sort of the primary use. Yeah, you'll actually find that a lot of, what's interesting is a lot of the medications that they'll give folks for migraines and headaches have other sort of psychotropic uses, right? Like for migraines, they'll give folks, oh man, the name just left me, but it's topiramate, which is an anti-seizure, originally started as an anti-seizure medication. The idea, I guess, is sort of, well, part of the problem is they give you these drugs and they're not really certain what systems they work on, and they're not really certain what system's causing you the problem, right? So what was causing your headaches? You know, what sort of brain circuitry or neurotransmitter system was not balanced just right. So they're like, well, we'll give you this drug that works on these particular, and see what happens. Some patients get better, you know, so there you go. Tricyclics, though, they actually inhibit the reuptake of norepinephrine and serotonin. We'll talk about serotonin here in a bit once we get to the 4th hour of class.
So they'll prescribe those separate though. Is there a reason there's a difference?
Prescribe what separately?
Like you can prescribe like SSRIs and SNRIs like separate.
Yes.
So what's the difference when they're in the same?
Yeah, I mean, a lot of it's going to depend on the constellation of symptoms and the things you're trying to treat, right? And sometimes you'll do things together. Again, tricyclics are not, you know, they're different than SSRIs or SNRIs. That's a whole separate class of drugs, actually, from your TCAs. And they're a much newer class of drugs that they also work on blocking reuptake, but that side effect profile is much different. And the effects are somewhat different as well, right? So they'll block that reuptake. But you might, it all depends on, again, the symptoms as you're trying to treat, which system you want to sort of work on. SSRIs are typically things you're going to give folks, SSRIs you're going to give them for irritability, aggressiveness, depression, things like that. Your SNRIs, primarily we're thinking about like Strattera, right, for folks who have some attention problems. Give them some norepinephrine. Now that sort of makes, I know it seems a little counterintuitive sometimes, right? Is usually like trying to think your way through this. And this is actually one of the first like non-stimulant sort of, right? Because they would also give folks who have ADHD like stimulant medications, right? So you might think like, that seems counterintuitive as well, right? Like why would if I've got some person who's already a little too antsy, why would I give them a stimulant? But the idea there is to like sort of activate their frontal cortex a little bit more than it probably is, right? So they're going to kind of stimulate activity there, and that like, hey, you shouldn't do that brain region, right? So that guy's usually taking a nap, and folks who are a little impulsive. And norepinephrine, again, I mean, if you think about this is a guy that gets keyed up. You know, it's used for that sympathetic nervous system, right? Kind of increasing vigilance, right? Again, that's not something that you might want to do to someone who doesn't have an attention problem, but if you do, that might help. Who loves cocaine? You don't have to raise your hands. That was bad timing.
I know.
I was like, oh, it actually is super powerful. In case you didn't know this, because it blocks the reuptake of dopamine, norepinephrine, and serotonin, which is an indolamine we'll talk about later. So it can block the reuptake of sort of like 3 neurotransmitters. So it's really powerful effects.
I have a question, it might be stupid.
You don't have to add that part.
Okay, well, it might be. Why can't you just test to see how much serotonin is being released to determine if they should get the different types of antidepressants?
Yeah. So that's actually a really great question, and I'll tell you what, it's hard to do. So there's a technique called fast cyclic voltammetry, right? And so what you basically have to do is to measure real-time release of a neurotransmitter. You've got to stick a super small probe into a small section of the brain. So you're not even going to be able to like do whole. You're going to do like a small section, and so you're going to have to drill a hole in somebody's skull. So right, we're already sort of hitting these thresholds. Now you can do this. And we do it experimentally. Folks do it all the time. They do cyclic voltammetry. You could also do dialysis, right? Microdialysis, so you could like sort of like suck out the fluid, right? And like that's in the synapse, like you could like pull that extracellular fluid out and see how much is in there. Again, that's very invasive, right? And you're going to get it at a very sort of. It's different if I'm doing this with a rat and I can force it to do what I want to, right? I can force it into a situation where I'm stressing it out or I'm making it feel right, and so then I can measure that real-time change in neurotransmitter release, whatever neurotransmitter that is. With a human, you can't really do that, right? There are these things like ethical concerns, and so if you're having some trouble with anxiety, for example, and I want to see, well, hey, you know, what's causing this? I'm going to measure your one. I have to figure out like what small brain region. And not even like, is it a whole nucleus maybe? It might even be less than that. Like what small cluster of cells do I want to measure? Okay. And then I'm going to have to drill a hole and go in. And then I'm going to have to do something that actually creates anxiety, right? Like, you know, in you. And so like, let's say you're like afraid of clowns. I don't know, right? I'm going to jump out dressed like Bozo the Clown and try to, and then I'm going to have to hide and hope that you can calm down from that, right? So I can measure those changes. So while that is if we could do this, and that would be great, right? Because then we would have a functional assay on what is actually the problem with this person. But experimentally, it's very difficult. Did that help? It is cool though, because you get this like you get kind of a current signature, and every neurotransmitter has its own, right? So you can see if it's dopamine or if it's serotonin that's being released or whatever it is, right? You can kind of get that cool signature, and it's great for experiments, but you know, it's some sort of like clinical setting. It's just not. It's probably not practical. All right. So we talked about reuptake. What do we want to talk about now? We want to talk about breakdown of catecholamines. There are sort of two major guys that we want to think about here: COMT and MAO. Right. So you probably have heard of MAOIs. Monoamine oxidase inhibitors, right? I think we may have talked about those briefly in class. Those are important. MAO and COMT both break down catecholamines, right? They sort of make these breakdown products. So dopamine, it's got a breakdown product. It doesn't really matter. Norepinephrine, interestingly, has a couple metabolites. One is going to be in the central nervous system, and the other is going to be in the peripheral nervous system. You don't need to know that. Okay. All those metabolites, though, they will go into the cerebrospinal fluid. Eventually, you'll get those into your bloodstream and then into your urine. And then you're thinking, well, can I just measure that in the urine, right? The problem with that is it's how many. This is a really personal question. How many times do you urinate in a given day?
Depends.
It depends. You should have said no pun intended. Yeah. So let's say 5-6 times. I don't know what's a good number. Right? Now I'm trying to think like how many times do I actually urinate in a given day? But let's say you go 6 times. I don't know. That seems reasonable. The problem is that like every time you go, and it's like half a liter of urine or whatever, right? That half a liter of urine contains many hours of metabolites, right? And they might not even be the preceding few hours of metabolites because it's been circulating in your blood and then it's getting filtered out. And so it can be, some delay in the timing there. So it's not going to be like this real time. Again, it's not a real time assessment. It works for things like if you're testing for diabetes, you want to see if there's a bunch of glucose in your urine. Like, well, that person has diabetes. But not so great for neurotransmitters. So MAOIs, your monoamine oxidase inhibitors, right? These again are antidepressants. You probably won't see a lot of folks on MAOIs. You shouldn't, hopefully. Again, because they have these sort of powerful side effects in terms of interactions with other medications. It's really what you're worried about, and dietary restrictions. So there are a number of things that you cannot really eat if you're on MAOIs, right? And you'll see this on a lot of medications, like don't take this if you're on an MAOI. You can also take drugs that, and these are also not terribly common, I don't believe, COMT inhibitors, right? You can take this sort of as an add-on to your L-DOPA, which helps with Parkinson's disease. So not only if you're taking L-DOPA, so we're going to increase that precursor, so you're going to make more, but we're also going to like sort of reduce the breakdown of dopamine, so what you do make is going to stay out there longer. So that's exciting, right? Don't worry about this. It's all these like brainstem stuff that you don't really need to know. There are these pathways. We've already talked about the nigrostriatal pathway that's involved in Parkinson's disease. Here we're just kind of showing that pathway for you. They're not a big deal. Ventral tegmental area, this is sort of important. There are two pathways that come out of here, the mesolimbic and the mesocortical, right? Mesolimbic obviously goes to the limbic system, so we're thinking about nucleus accumbens and so forth. And the mesocortical is going to go up to your cortex. These guys are involved in like reward and learning, memory, things like that, and drug addiction as well. So that's the VTA to the nucleus accumbens. And here's the VTA to your cortex. Also, as I said, learning and memory heading out there to your hippocampus. Why does this stuff keep going up to the olfactory nucleus? This is in a rat. Rats navigate their environments using their olfactory senses, right? They're primarily olfactory and somatosensory. They have the whiskers, and they have a thing called the barrel cortex, because they have barrels that they have on. No, it's shaped like barrels. If you stain, like the cortex has this barrel-shaped pattern because of the way the whiskers are set up. So something terribly important for you, but it's sort of interesting all the same. So it's going. A lot of this is going up to the olfactory cortex as well. Some of this also
I have
goes.
a question. You were saying that basically the mom doesn't love you, she's just addicted to taking care of you. So, in, I guess we'll just say mothers who neglect their children or like abandon them or just leave them, is something wrong with their VTA?
I have no idea.
Okay.
That's a good question. You could check that out. So now I'm thinking of like an fMRI study where you're going to put like regular mothers. You have to have like 3 groups, right? You've got to have those like. So you ever want to like, and you want to talk about something, and then I want to say, well, maybe like spoiler alert, but I don't know. And if I actually tell you spoiler alert, and then I actually like mention any of it, then it's going to ruin it for you. So I'm just not going to say it. So you have to have like 3 groups of people, right, Cassie? You'd have to have, but probably 4 groups, right? Because well, that would work. Never mind. I was going to have like non-mothers in a group, right? So you might want those. But you would want like what you would identify as regular mothers, like doing normal mother things. You'd have to have a group of neglectful mothers, and then you would also want a group of like overprotective mothers, right? So you really want, and then you, because they may have like extra activity going on in their VTA and nucleus accumbens, right? That'd be kind of exciting. So why don't you work that out?
Right.
I'm excited to see what happens.
If the department would get an fMRI machine.
Yeah.
Yeah.
Well, somebody around here's got to have an fMRI machine, right?
I see a lot of neglectful mothers, that's.
Convenience
That would be
sample.
easy to find.
It's a convenience sample, right? We just need an fMRI, we can do this study.
It makes a lot of sense when you think about the moms who have substance use disorders, right? Because they're getting that dopamine from somewhere else, so they're not, like, that's not a need that's registering.
Yeah, it's brilliant,
They
right?
don't
It's a
need that
brilliant-
kid
Huh?
to feel better.
Chemically,
They don't need the
they
kid to
don't.
feel better.
No, why'd they even have the kid?
Accidents happen.
How else would you save your marriage?
I thought you guys were supposed to help people with that. So, yeah, I think, Corey, that's actually a pretty good observation. If this guy, I mean, think about those, kind of like those mice that don't have dopamine autoreceptors, right? They don't even know they're on cocaine. They're on some other drug. They don't even know, like taking care of their kids, not really adding anything here, right? See, that's a whole, now we've got a whole spin-off study over here of not just neglectful mothers, but drug-addicted mothers, right? And then we could really like describe some things. You guys are onto something. I'm sort of impressed. I wasn't expecting anything worthwhile. I wasn't expecting any sort of research insights, right? You guys aren't interested in that stuff for the most part. Right, Jason? You're going to have to do a research project, or at least a fake one, right? How many of you are going to do the fake one? That's disappointing. I had high hopes
No,
for some.
Kristen, you did a fake one, but you ran it.
Yeah, but I don't want to write a thesis, that's like hard.
I think it's the same. You guys should all write the thesis. That's what I encourage people to do. It looks better. I mean, the other one's just like a proposal. You can propose anything, right?
Are you gonna fund it at first?
No,
And help with the sample. and all that.
possibly. I would do that.
Buy an fMRI machine.
Yeah.
That one's gonna be tough.
Get a grant.
But you know, there are probably people, I don't know, does the hospital have an fMRI?
Oh, no,
Probably
they're
not.
just gonna lend out.
No, but you can pay for hours on it. And then if you find someone who has access to it, who really needs like some help with like a research, you know, like they need to add something to their career, like a research project, then you're onto something.
What money?
They can figure that out. Like that's, you know, you've got to have a money man and an idea man, right? You needs to be the idea person. Let them be the money people.
Just sneak in. It's fine.
Just boot that thing up on your own, right? When I was in grad school, we had a, no, not in grad school, I'm sorry, when I did my postdoc, when I was at Wake Forest, they had a Meg machine, which was like it's a giant magnet that measures the changes in the magnetic fields in your brain. Yeah, this is pretty awesome. And the magnet was huge, like they had to build an add-on to the hospital to put this thing in. And the walls around it were like this thick, like the door literally was this thick. I knew that, like, because if you went in with just like the smallest piece of metal, it had to be so inside, like even, you know how the center of the earth is made of iron? That would even screw it up if you didn't have enough, that, like seriously. And you had to have that much sort of like insulation between them. It was pretty impressive. They didn't let me use that. I wasn't that, it was, you had to put people in there. I wasn't too interested in that. I didn't like to put people in there. What else do we need? We've got Parkinson's disease. We talked about that. Give people neurotoxins. That's interesting. Don't worry about this. This is your striatum, though. By the way, if you're a rat. Now, this is not how your striatum probably looks, because it's again a rat. This is the rat cortex coming around. We've got those genetically engineered guys, knockout mice. Don't worry about this. Different genes. There are dopamine deficient mice that you can use for studies. I really just want you to kind of be familiar with this in case you come across something later. Again, if you hit these folks with L-DOPA, these mice, they can actually restore some of their behaviors that they have, right, because of the lack of dopamine. There's your VTA. We talked about that a little bit. Again, this is that reward pathway, right? If you get your medial prefrontal cortex involved, that's the guy that like tells you probably shouldn't do things, right? So they can get a aversion involved. Part of the problem with kids, I don't know if you guys have ever seen kids. When they're born, they have this thing called the amygdala, right? And you've got one too. And it's the same as when you were like 3, okay? But what changed in you was your medial prefrontal cortex, right? When you were a kid, this guy wasn't really online just yet. And whenever you went to the store and you didn't get, I don't know what, like that My Little Pony or whatever it was that you wanted, right? I don't know what you guys played with. And you thought, well, that's the end of my life. and I'm gonna just like pitch this total fit, right? That's because your amygdala was all geared up, and your medial prefrontal cortex was like, not ready yet.
That's why kids are such assholes.
Yeah, intentionally, right? I remember when I first moved here, I went to, but then I think sometimes it's their parents. Because when I went to Walmart, I moved here and I had to go to Walmart like, it was like at midnight or whatever, right? And it was like an emergency thing. I was like, normally I try to avoid Walmart if I can. But I was like, I absolutely need, I think it was like dog food or something, right? Because I found this dog in the middle of the road. I know, right? It was a great dog. He died, but.
Did you keep it, or did it die like right
No, I
after you
kept it.
found it? Oh.
I kept it like a year and a half, and he died. He had tumors. I think, yeah, I don't know. I think the people abandoned him, and then they tried to take him back from me, and I didn't give him away. That's a whole separate story. So I got him in February, like after I moved here in like the end of December. So in February, like 3.5 years ago, we had this big snowstorm, right? But before that, I found this dog. I was just driving home from work, and my wife said, hey, there's something in the middle of the road. He was literally running down the yellow lines, right, down the middle of the road. So we get out, and I pick him up, or she picks him up, and it's like a dog. So that's something, right? So we ended up with a dog that way. And so we got him all cleaned up, vet, everything. It's February, it's snowing, and I'm taking him out to go to the bathroom, right, because he does that outside. And where we lived, there's like a bridge, and then a bunch of like power lines were down because apparently they had to replace like 8,000 electrical poles. So I picked the dog up because he's like this big, right? I was like, I'm gonna carry this dog. And these people like circle around twice and look at me. And then they pull over and they ask me like, hey, is that dog new? This dog's old. And this dog's at least like 10 years old, right? And you can just tell because he's old. I was like, oh no, I've had this dog a long time, like 6 or 7 years. I'm not giving him back to those people. He didn't. He like took his head and like tucked it and hid away from him when they drove by. When they stopped. Yeah, right? He had like 2 growths on his neck where they'd had like one of those like shock bark collar things on him. Yeah, we had to have those removed. I know, see, we wouldn't have given him a dog either. Forget that. But then he died. So he had, I think he had tumors anyway, so we took him to the vet. I picked him up one day and he like really screamed at me. I was like, I'm taking him to the vet tomorrow. I don't remember why I told you that story.
You
About
went to
that.
Walmart.
Yeah, so, thanks Sierra. And there was this little girl like screaming at the top of her lungs, I'm about to poop my pants. Like repeatedly, I'm about to poop my pants, I'm about to poop my pants, I'm about to poop my pants. And there were like 3 adults with her, and I thought, that's like the nuclear option when you're a kid, right? Like if you scream, I'm about to poop my pants, your adult, whoever that is with you, has to take that seriously. That's not it's like, I'm so thirsty, I'm hungry. Some of that you can ignore if you know we just left McDonald's, you cannot be hungry or thirsty. You can ignore that. But the I'm about to poop my pants one, that's the one you gotta take. But they were just like in the chips aisle, like buying potato chips. And this girl's just like screaming, I'm about to poop my pants.
Maybe she had a diaper on.
If she had a diaper on, she was like 4 years too old to be wearing it.
We watched a video yesterday of a teenager being bathed by his mom, so nothing would surprise me.
He was not a teenager. He was like 7.
He was too old for a bath.
He was
Approaching.
too old, but
He didn't need that fake beard. He was coming in there.
That's weird. Is that what you were told? That's weird?
That's what we said. That's weird.
Yeah. All right.
I have a question about the brain. Okay, so I work with.
Does that help you?
Yeah, I work with kids who have experienced some different traumas and things.
Yeah.
So I'm wondering, like with them, like a lot of times they don't have like impulse control and like they do things and it's like they don't understand the consequences of their actions. Is it because like the front part of their brain hasn't, like something has stunted that and they just can't do that? Or is it the amygdala has been so altered by early childhood trauma that that's just overpowering anything that the prefrontal cortex could do? Does that make sense?
Yeah, so that's a great question. I think it's a complicated question because the, I'm imagining, it's just my assumption, based on my experience with this. Kids who have experienced early childhood traumas, those ACEs, right? Is that what they still call them?
Yeah.
ACEs, you didn't know I knew that. I'm impressed with you. I think my assumption is, not only, if you have a lot of ACEs, there's probably a pretty good correlation with other sort of issues that would cause you developmental issues, right? So then you have to worry about nutrition, the prenatal environment. There are other concerns, right, that could cause developmental issues and problems. On top of that, you do have that overactive amygdala, right? And that amygdala is always working kind of from the beginning because it is that fear nucleus, and you really need that guy to be active. And it can be activated early, and if your earliest experiences are going to be those traumatic experiences, then it is going to be much, much more likely later to cause you problems. Folks, there's probably one of the best correlations you'll ever run across between ACEs and the likelihood that you will develop PTSD as an adult, right? Having, even just one or two sort of traumatic events as a child drastically increases the likelihood that you're going to develop PTSD as an adult, right? And a lot of that's because you're right, that amygdala circuitry is already geared up. And then there could be some developmental issues where that prefrontal cortex isn't coming in online. Now, the other thing, you say you work with kids, your prefrontal cortex really, we're even talking maybe 12, 13 years old before it's really fully online. So anything before then, you're still dealing with, even in a child with no traumatic experiences, still dealing with an underdeveloped prefrontal cortex.
Right, yeah, I'm just thinking about it like in response to sexual trauma as a young kid, and then like as a teenager starting to have inappropriate sexual behavior, and then if that continues into adulthood. Because what I'm doing my dissertation on is female pedophiles, so I've been reading some different things about all of that, but then I see that also with some of the kids I work with too, so then I'm just thinking about how the brain could potentially be altered and what's actually going on.
Yeah, I think that's interesting. You still need that fMRI machine.
Yeah.
I guess that's a good project. I like the other one, but if that's the one you're kind of committed to.
The other one could be the backup.
Right. Female pedophiles, huh?
Yeah.
Yeah, I'd say there's not a lot of literature on that, is there?
No. But what I found is interesting.
Yeah, no doubt, but like the two papers, that's what I would think, right? I don't know. You typically don't think about.
Exactly, which is why there's no literature out there.
It is, yeah, it's kind of interesting. It makes me think about that whole bathroom debate they had, I guess it's still going on, right? I walk in the one with the orange door. By the way, first floor, it's an orange door as well. I don't know about the second and the fourth, but the men's room on the first floor is an orange door. So I can tell you that, I can confirm that. They were talking about let's ban, whatever subgroup you want from bathrooms to prevent sexual assaults in bathrooms, right? And then there was I read this thing, I was like, well, you should really, statistically speaking, ban, straight white cisgender males, because they're the most likely to be, statistically speaking, most likely to be sexual offenders. And I thought, well, that's kind of interesting. And I hope it doesn't go there, because I do need to use the driving home would get inconvenient. But there you go. I don't know if that helps or hurts, but that's a story.
That helps, yeah.
All kinds of different subtypes of dopamine receptors. We've got 5. They do different things. Remember when we talked about inotropic and metabotropic stuff? So don't get too worked up about that. Do keep in mind the D2 or autoreceptors. That's sort of important, but nothing too much to worry about there. I'm not going to hit you with these diagrams. I really just want you to think a little bit about dopamine. We did talk about that potassium channel business. not a big deal. This is interesting. Apomorphine, this guy works on D1 and D2 receptors, and you actually get a lot of the behavioral activation that you would see with cocaine and amphetamine. And there are all of these sort of things like you'll never, like, you know, you're not gonna see somebody taking SKF 38393, right? And that's not a label. That's not a medication. These are like experimental things. What else do we want to think about? If you take too high of a dopamine antagonist, right, this is bad. You can get catalepsy. This is when you sort of just like stop moving. That's bad, right? So we don't want to screw around with this too much. How many of you have heard of haloperidol? Yeah, right? You get a couple head nods there. That guy is a D2 receptor blocker. So if it's going to block those D2 receptors, what's that going to do? That's the autoreceptor. If we block the autoreceptor, we're going to increase dopamine, right? That's a cataplectic rat.
He's a cute caterpillar.
He is. Rats are cute when they start out. I had a baby rat for a while. It died.
Did you experiment?
No, I had it when I was a kid. My neighbor brought it over and was like, "Hey, look what I found." And then I
It was
convinced.
like a street rat. Or like a pet rat.
It was like a field rat.
Oh my god.
And I mean, it just, you know, my neighbor found it in a field and he brought it over, and I convinced my mom it was a mouse, which she found more palatable than a rat, and let me bring it inside and like feed it puppy formula. I tried to take care of it, but it just, a single rat pup without a mother, you know, it just didn't work. I had a turkey pigeon once too. That was a crazy one.
What's a turkey pigeon?
It was a show pigeon. That one just showed up at my house when I was a kid. It was like this purple, and it had these like feathers, its tail feathers were up like turkey feathers.
And your parents just let you have these things?
They just showed up, so they had to. I don't know, that was their logic. Yes.
There was no farm to take them to?
I
Like, quote unquote, take them to the farm.
No, I think.
Yeah.
A turkey pigeon, it's a damn pigeon.
They weren't just murdering his pets in the back.
No, I think the reason, because there were like farms all around us, that like taking it to the farm would not really work, because across the street, like why are you taking it over to Marvin's? That doesn't make any sense. Just leave it here. So I don't think that really works. I grew up across the road from a goat farm.
Oh.
Yeah.
I love goats.
Yeah, he still has a bunch of goats, and my folks still live there. Dopamine. You guys need to know something about dopamine. The genetic engineering stuff. I mean, there are like a ton of knockouts, right? That we can think about. Dopamine transporter knockout mice are hyperactive. That makes sense. What does the dopamine transporter do? That's reuptake, right? If you have no ability to reuptake dopamine, guess what happens? Yeah, you're active. You're hyperactive, right? Because dopamine influences that, right? They also have a bunch of cognitive deficits. They're impulsive, and they kind of act like kids with ADHD. So that's interesting. Don't worry about this too much. What's a wild type mouse? That's just a regular mouse that we've not screwed around with its DNA. Just to give you a heads up there. That's wild type. Homozygous is when we've screwed up with both copies of the gene. Heterozygous is when we've only screwed up one copy. Okay? So if you kind of see that language out there. Okay? So in this case, what you see, and this is measuring hyperactivity, like how many times does it move across a light beam, right? So we're going to measure that. And that's a measure of hyperactivity, how much is it moving. Here's the wild type. Here's the one that's heterozygous, so it's got like one good copy of the dopamine transporter gene for its DNA. Here's the guy who has like 0 copies, right? And you can see how much more active it is at every single time. This is when the mouse is getting tired. You know, 180 minutes, I mean, we're 3 hours into it, so. That's an active mouse, right? Like 400 times within the first 20 minutes, it's running back and forth across this cage. I mean, that's really what this is telling us, right? That's a pretty hyperactive mouse. Knockout mice. Here's an uplifting story. If you don't have D1 or D2 receptors, these mice die in about the second or third week of life, so they don't live very long. Again, they're used to study psychoactive drugs. That's why we do this. Just showing you differences. I'm not going to get you too heavily on this. Now this is sort of interesting. So if you continue to give someone, like haloperidol, for example, give it to these rats for a long time, they develop behavioral sensitivity, right? So that's kind of interesting. And the sensitivity is due because they're sort of upregulating their D2 receptors, so they're increasing the number of autoreceptors, right? Because haloperidol was doing what? It was blocking D2 receptors, right? And so they're not getting stimulated. So they're continuing to release dopamine, and you're like, wow, I'm not really realizing I'm releasing dopamine. How am I going to fix this? Well, those autoreceptors must be faulty, so let's send some more out there. So you'll upregulate the number of, you'll increase the number of autoreceptors that you have.
Is that Haldol?
Yeah. So we talked about dopamine. We can talk a little bit about norepinephrine and epinephrine. This is going to be a shorter conversation, largely because the only thing there's only like sort of one drug that you might think about that works on the norepinephrine, and that's like Strattera, right? It's going to block the uptake of norepinephrine. But there's some other things we might want to think about. Your noradrenergic system is pretty widespread. Starts out here in the locus ceruleus, which means the blue region. That's interesting. That's why they put it in blue. I don't know if that's why they put it in blue, but they should have. So we'll give them some credit for that. Again, this is going to work on your sympathetic nervous system. That's why we also call it adrenaline or noradrenaline, right? So if you think about an adrenaline rush, and what adrenaline does for you and how this system works, you can think about the things that are mediated by norepinephrine, right? We like to say it's the 4 Fs. What are the 4 Fs? Feeding, no, not feeding. Fighting, fleeing, freezing. Is that the other one?
Oh, I said feeding, fighting, fleeing, and fucking.
Yeah, I usually get the last one. What was it? Feeding? I don't think feeding's on it, right?
Oh, I don't know. That's
Maybe
you said
it is.
4 Fs, and that's what I've always remembered. Feeding,
We'll
fighting,
go with that then.
fleeing, and fucking.
Yeah.
So, but freezing is probably should
I don't-
be added on there.
Not digesting. That's the thing I want to be clear. Not
Yeah.
digesting, right? So not while you're digesting the food is norepinephrine going to be involved in that. Actually, it's going to stop it. All right. Fight or flight. We got that. Not a big deal. Blood vessels. Again, a lot of receptor subtypes, right? You've got 2 alpha receptors. These are alphas, by the way, in case you're not familiar with alphas, but you should be seeing alphas, right, from like stats classes. And then this is beta. Not a big deal. A lot of these guys use second messenger systems, right? Those are the metabotropic things that we talked about with like cyclic AMP and so forth. Let's see. A lot of it, again, arousal, cognition, consolidation of memories, emotional experiences. What are some other things we should think about? Again, it's involved in wakefulness. If you, kind of reduce norepinephrine, people get sleepy. That makes sense, right? Nobody has had an adrenaline rush so powerful they fell asleep. That doesn't happen, right? That's not how adrenaline rushes work, so you can kind of keep that in I don't know, right? Who knows? Hey, who loves to enhance their working memory? All right, well, you can take some drugs for that. There are drugs that are going to be, they're going to activate those alpha-2 receptors out in the prefrontal cortex, right? So you can really key up your working memory. There you go. So a graph showing that excitement. Cognitive tasks. Again, if you go back to this, let's find it. I mean, look at how much it innervates, the system innervates the cortex and your hippocampus. I mean, it's getting hit with the hippocampus like 2 different ways here, right? And so you're really, that's going to be the learning and memory section, working memory, cognitive capacities, all of that. You can see where that's going to be affecting. All right, let's see what else do we need to do. There's your working memory. Keep in mind you don't want to get too amped up, right? How many times have you been on such a high adrenaline rush you forgot what you were doing, right? You don't have memory. This is one of the reasons folks in stressful events don't have great memories of the events that happened to them, right? If you're asleep, you obviously don't remember what happened, right? But if you're so amped up, you're not going to remember either. So you want to find that peak, right? What's that peak balance there of your norepinephrine?
So what's happening with Strattera when like, because it's I understand it's commonly used for ADHD, and anxieties, kind of like COVID. So what's happening there to keep it from like ramping up the anxiety like a stimulant might?
Yeah. Well, I mean, its activity is, it's going to block reuptake of norepinephrine. So we're not massively sort of like increasing the production of norepinephrine. We're just going to block its reuptake, so it's going to stay out there longer, have a little bit longer effect. It's powerful enough to have an effect, but a lot of it's dosing and the potency of the drug, right? So you want to make sure that you're not. It's the same reason, like why do people take caffeine? Like why would you drink coffee to stay awake instead of using amphetamine, right? They're working on similar systems, but they have different potencies. Mm-hmm. So Strattera is good, but not too good, because that would be a problem, right? You don't want to get somebody so far over, right, that you're going to actually cause some anxiety problems, because they're already going to have some issues. Memory consolidation, you guys don't even know exactly how this trial process works, but it does work. And epinephrine is definitely involved in that. Let's see. One of the problems with epinephrine is it increases blood glucose levels, right? Which is not necessarily one of the problems, that's one of the ways it might actually work, right? Your brain definitely needs glucose to do its thing, right? And so by increasing the amount of glucose that's there, it sounds pretty good, right? But gotta watch that. If you already have some diabetic issues. Also, epinephrine, norepinephrine, they have a lot of peripheral effects in the heart, blood vessels, other places. You can see which receptor subtype is going to be activated and sort of that effect. These are side effects, things you would have to worry about if you're giving someone a alpha or beta agonist, right? That was to treat an issue with, like an epinephrine agonist or norepinephrine agonist. You'd have to worry about causing some of these other problems, right? So, especially cardiac conditions, got to worry about that. If you think about that adrenaline rush, right? If you're constantly having that adrenaline rush, that can be problematic. So watch out. They do use it for asthma. I don't think you guys really care about asthma or decongestants, right? Well, you can take albuterol. There you go. Have fun with that. There are drugs that treat hypertension. Again, you can see that those sort of peripheral effects, things that I don't think that you're going to be dealing with. I mean, some of your alpha-2 agonists you can combine with sedatives, other things, right? Whatever you want to do. There aren't a lot of things here that I think you need to think too much about. But here are some drugs that affect your noradrenergic system if you need to take a look at that. I'm not going to ask you a whole lot about that. I don't think you're going to see folks in that situation.